So I'm with Tracery Ophthalmics, and we're harnessing artificial intelligence for precision drug development in blinding eye disease. Oops. And so, I'm going to focus on dry age-related macular degeneration. And as you know, most of our patients have no treatment. And this is because drug development is imprecise. We know the disease is complex. There's over 200 polymorphisms. The disease is profoundly influenced by whether you smoke. We can't capture that kind of complexity, especially when you look at the way that we group our patients. We tend to say early, late, wet, dry, intermediate. And the only thing we can measure is a patch of dead tissue. And that means that right now our trials are late, they're large, and they're costly. Basically, we can't figure out which patient to put in the right study. So Tracery's, our goal is to get the right person into the right clinical trial targeting the right molecular pathway with the right chemistry. And so we apply artificial intelligence to each of those three pillars. So we apply artificial intelligence to our own imaging method that I'll show you. We also apply it into large data handling. And then finally, we've actually partnered with an in silico chemistry company that's helping design molecules that match the pathway that matches the person. And part of this platform is not only integrating it, or it's not just applying it vertically, it's actually integrating it horizontally. So what we have is we have our own proprietary image acquisition process. We've developed our own convolutional neural networks. We have our own prospectively acquired curated databases, and we've got partnerships with our own drug development company, which is TMI that you heard about, and also with Cyclica. So I'm going to show you a little bit of our imaging technology. So this is what we currently look for in dry AMD. This would be a patient with a patch of dead tissue. When we use our own imaging, we can clearly see more disease. And the interesting thing is I'm a retina specialist, and I've seen now thousands of these pictures, and I feel like I'm looking in the other end of the Hubble telescope because I'm seeing such new and interesting things. This is a functional imaging method. We see entirely new complex phenotypes. So basically, these are new flavors of patients. So they're, again, fundus autofluorescence. The periphery of this patient looks pretty normal. And when you use our imaging, you can see much more disease, new patterns, and you can certainly confirm that this disease extends beyond the macula. So this addresses the heterogeneity. We also have prospective data. And so with prospective data, it means we can go back and look at it, and that permits predictive modeling. With these new phenotypes, now we can do phenotype first correlations with genotype and ask, well, if they look like this, what's their molecular biology or their smoking habit? We can, of course, do it the other way around. We can now say, well, in people in that particular pathway, maybe they look this way. And of course, once you know the molecular pathway, then you can pick your targets and develop your drugs. So I want to show you, this is how we started with our image analytics, and it's quite funny. I can tell you, you can't chain graduate students and retina fellows to the table long enough to do this with all of our images, because we were tracing out these patterns. These were handcrafted feature extraction. So what we did was we got ourselves an engineer, and so we've been developing our own convolutional neural networks. So we've been training these. I will say we actually started training on the color using 2 of the Google platforms as well as AlexNet. We've done cross-modal training. So these are just 3 layers from one of our algorithms, and you can see the machine thinking about the picture, bringing it together. We also do this sort of clustering analysis that you were talking about, the hierarchical feature representations and dimensionality reduction. My fellow said one day to start lab meeting, he said, you know when you're working in 30 dimensions? No. So with all that in mind, he reduces the complexity of these images such that we can classify into new phenotypes, allowing us to address heterogeneity. And of course, again, going back now, by having prospective data that we can go back and look at, we can turn a classifier into a predictor that should be able to, we're hoping, will be able to tell us who's going to get GA, who's not, who's going to get CNV, and who's not. Ultimately, add that to everything else about the patient for precision drug development, and ultimately for personalized medicine. So we are, we're a JLabs company. I will say we won a top award globally for artificial intelligence for drug development. We've won, our funding is all entirely non-dilutive. We're looking for additional patient cohorts, correlates with omics data, want to advance our analytics and expand through the cloud. This is our team, and of note, the two people on the right bring extensive AI background to us, including imaging and chemistry. So Tracery's goal is to get the right person into the right trial, targeting the right pathway with the right chemistry, and we vertically and horizontally integrate that to provide data, information, and answers for precision drug development and personalized medicine. Thank you.
Great. Thank you very much, and thank you for staying on time. We have time for questions here.
Shelley, thanks. Just interested in the size of the data set that you've been performing your algorithmic work on thus far.
Yeah, so, and this is a really important question. So our data set, unfortunately, it's just me. We've been working on it for a number of years. So we actually have just around 500 eye sessions, and from those we have multiple images. And so we have about 32,000 images that we use, but the key was training it, cross-training it on other neural networks, and then from there doing cross-modal and multimodal analytics. So our dream is that we could start working with people who have large cohorts and prospectively move forward. Because if we could do this, I think I'm taking away your question. Because I mean, it's interesting to me. I mean, biomarker development, just like drug development, biomarker development has, even digital, has taken 10 years. And if you go back to fundus autofluorescence, from the time it was first introduced until it got incorporated into a clinical trial was a decade. We believe that if everybody did this at once, we could do it in 2 years. And that's the value proposition, is bringing a new tool forward in 2 years rather than 10.